So my name's John. I'm based at Imperial College, and I'm going to be talking to you today about some research I've been doing as part of my PhD on environmental drivers of extinction and diversity in crocodiliforms. I just want to start by saying thanks to NERC for funding this as well, and I guess to my supervisor for being excellent. So as we all probably know, crocodiliforms are an extant group of pseudosaucian archosaurs, definitely the coolest group of archosaurs around. And they have a very rich evolutionary history. So if you go back in the past, there are hundreds of species, and they're way more diverse and far more disparate than they are today. And if you go back to the Jurassic and Cretaceous periods during the Mesozoic, then we see loads of diverse forms which are not really around anymore. Like we have two radiations of fully marine forms. So like these guys are actually swimming, and they actually have convergent evolution with things with other marine reptile groups. And during the Jurassic, we see that often, or most of the time, crocodiles were confined to kind of temperate latitudes, with the equatorial zone being seemingly devoid of crocs, and especially around polar regions. But if we move into the Cretaceous, we see that they have a more global distribution, even reaching kind of the polar regions down in Australia down there. So while they have this really kind of nice evolutionary history, it's very rich, it's full of many radiations and many apparent extinctions, very little is actually known about their kind of quantitative history. And we wanted to investigate this using some fairly new methods and protocols, and in particular focused on the Jurassic-Cretaceous boundary, because it seems like there's a lot of important stuff going on around that time. So this is just like a pretty standard raw taxonomic diversity curve, and if you look at it, then it seems like there's a pretty hefty extinction going through the Jurassic-Cretaceous boundary, marked on there by the red dashed line. But as we all know, you can't take the fossil record at face value. There are many ways in which we've sampled it which influence and bias the way in which we reconstruct these kind of curves, including a lot of really great research done here. So we wanted to see, is this diversity crash a genuine kind of biological signal, or is it an artifact of the way in which we've sampled the fossil record? And as well as this, we wanted to look at differences between the terrestrial and marine realm. So as you can see, there's a pretty hefty apparent decline in marine forms again. But when I say marine throughout this talk, I mean fully marine, so like fully pelagic, open swimming crocodiles. It's a bit weird to think about, but they definitely existed at some point. And by terrestrial, I also mean include shallow marine forms, such as those which dwell on coastlines, as well as freshwater taxa. So what do we want to know? We want to know, is the JK boundary decline real, or is it a sampling artifact? And if we use different methods for accounting for sampling biases and stuff, do we get the same signal? This is kind of important, because quite a lot of the time people use something like residual diversity estimates, and they don't actually test whether that's the same result as you get if you use like an ecological subsampling approach. As well as that, we wanted to investigate what was going on between the marine and the terrestrial groups, and if we're getting different signals, why? And as well as that, look at the environmental drivers of the extinction signals we're seeing and the diversity patterns. So first off, this is a phylogenetic diversity curve, and this is based on a newly constructed informal supertree consisting of some 271 species. So it's not built on the Branzati et al. 2012 supertree. This is brand new, constructed firsthand, very painstakingly. So phylogenetic diversity is diversity calculated from adding on the in-bin occurrences of taxa plus ghost lineages, which are lineages inferred by where we know that a taxon must have existed, but we haven't sampled it based on its sister taxon relationships. And then you sum all that together and you produce a nice little curve, and then you can resolve polytomies and uncertainties in various ways. But no matter kind of how we kind of played around with the tree, we always seem to get the same signal, but you actually have this huge crash in diversity going through the Jurassic-Cretaceous boundary again. And this is actually kind of weird, because normally when you do calculate phylogenetic diversity, you get a kind of back-smearing effect due to some uncertainties in the topologies, and it's very difficult to actually get a decline of that magnitude. So we're fairly confident that what we're seeing through the JK boundary using phylogenetic diversity is a genuine artifact, that this decline is definitely being picked up. As well as that, we divided it into the marine and the terrestrial groups again. And what we see is this kind of decoupling of responses through the JK boundary. Both marine and terrestrial fauna seem to suffer a decline of some degree. But while terrestrial taxa seem to recover fairly rapidly, marine forms actually never do again. And this is due to the extinction of Thalattoseuchia, so the main marine group which goes extinct in the Valanginian Hotarivian, so the very earliest Cretaceous. Then after that, we don't actually see much going on. Like, we have radiation of new groups, but they always seem to be suppressed in their total diversity. And I'm not quite sure why this is. It could be due to the radiation of other marine reptile groups such as plesiosaurians getting really diverse around this time, or it could be due to some kind of extrinsic environmental factor. If we use SQS, so I'm sure everyone loves SQS and subsampling protocols. SQS is similar to rarefaction in that you subsample based on lists of taxa, except that instead of instead of rarefaction, where you kind of subsample to a kind of fixed threshold, this is based on the Goods-Mu estimator of coverage, which is a kind of estimate of how well sampled your taxa is, and then you kind of use that to set the threshold of what you're going to subsample to. And this is what you do is you set different quorum levels based on this estimator of Goods-U and coverage and evenness of sampling, and then you subsample based on that, and then you produce a diversity curve, which isn't like actual diversity, it's proportional diversity. So what really matters here are trends, and as you can see, like if you assume that you've got like really good sampling going through the Jurassic Cretaceous boundary in marine crocs, then you actually get a decline going through the boundary there. So it's not as big as the phylogenetic diversity was picking up, but there's certainly a decline there. Unfortunately, most of the rest of the early Cretaceous is too poorly sampled to reliably produce estimates, so I've just put like a crocodile there because there's no data. If you look at terrestrial forms, you do pick up this huge decline going through the boundary again, and this is again assuming fairly good sampling going through the boundary. This is actually almost on the level of mass extinction status. It's about 85% of taxa go extinct. But as you can see again, we have this recovery going through there as well. If you look on the kind of spatial scale, so this is subsampling again using SQS on the continent level, then we see huge declines in North America and Africa going through the boundary, whereas European forms actually seem to be okay. Asian forms decline right up to the boundary, but then in the early Cretaceous, after this decline, you actually pick up a radiation in South American and African taxa. So it seems like we're getting different signals here from Gondwana and Eurasia and North America, whereas most of our crocodiliform knowledge is coming from the northern hemisphere during the late Jurassic, but then something happens, and then all of a sudden, in the early Cretaceous, most of what we know is coming from Europe and Gondwana. If you calculate extinction rates as well and origination rates, so this is using the 3-timer extinction and origination rate, but if you use boundary crosser estimates too, then you get a very similar signal. I just couldn't be bothered to put on another slide. But you see extinction rates are highest in the latest Jurassic, and then they decline through the early Cretaceous. And origination rates as well seem to decline at the, seem to be very low throughout the, so origination is speciation essentially, but it's very low through the early Cretaceous. And so what we're seeing is very high diversity during the latest Jurassic, coupled then with massive high rates of extinction coming up to the boundary, and then depressed origination for some time after that, and combined with low diversity as well. So that's just like the general kind of like trends in what's actually going on in terms of biodiversity and extinction. But just by itself, that's not very interesting. So as well as that, we looked at the environmental drivers of these. So we got quite a lot of time series data from the primary literature. And as with you're supposed to treat these kind of things, we took the residuals of these data using a first-order autoregressive model to account for serial correlation between the time series data, and then we just used some basic correlation tests. So we calculated things like the Akaike weights of each variable with the various diversity and extinction metrics, and as well as that, we just performed basic correlation tests. And we were fairly rigorous. Like we said, like for an environmental parameter to be associated strongly with one of our diversity metrics, it had to be significantly correlated using 2 different measures of correlation, as well as be 4 times higher in the Akaike weight than any other possible variable when we were doing these model fits. And what we actually find is that, like with raw taxic diversity, as we might expect, the patterns which we're getting seem to be driven largely by proxies for the rock record. So I know that Mike up there is probably thinking, you can't use proxies like formation counts and non-marine area, but they're the best we have at the moment on a kind of global scale. And it seems that diversity, when you look on a very raw scale, is driven mostly by these proxies for the rock record. So, it says sampling is important. We have to take into account these kind of things. Which It's actually quite nice because there was a paper on crop diversity fairly recently which just decided to ignore this and somehow got into Nature, despite being fundamentally flawed in these kind of aspects. But when we look at the kind of subsample data, like the corrected diversity curve, we actually find that things like sea level and the strontium isotope curve seem to be responsible for a lot of marine diversity. So they seem to be driving these patterns. And this is fairly consistent with what's going on kind of tectonically at the time. So Gondwana and Laurasia are undergoing like full-on fragmentation at this time. We're seeing like the fastest rates of seafloor spreading and continental fragmentation and like tectonic movement going through the Jurassic-Cretaceous boundary than we see for like the rest of the Mesozoic. And it's just like maybe it's these large-scale patterns which are actually driving these diversity patterns in crocs. In terrestrial diversity, though, we see instead it's sea level as well seems to be playing a role. But as well as that, it's atmospheric sulfur toxicity seems to be the key driver here, which is contradictory to a recent study which showed that in fact it was probably temperature which was driving crop diversity through time. And this is actually kind of cool because like at the Jurassic-Cretaceous boundary and through the early Cretaceous, we actually find that we have several of the largest igneous provinces emplaced that we see pretty much in history. So in the southern hemisphere, we have the Paraña-Ettendecker traps going off in the kind of Hota River in Biremian. And as well as that, we have the Java-Otongan Plateau. And these volcanic provinces are like the biggest we pretty much see throughout history. Being kind of largely overlooked, actually, in terms of like their environmental influence and their potential impact on diversity. But it seems like maybe these guys were spewing out massive amounts of sulfur into the atmosphere and causing havoc with crocodiles. So not good, not a good time to be a crocodile around this time. So just to kind of like bring all that together, we do see this huge diversity crash in crocs going through the JK boundary, and this seems to be a genuine signal because we've applied two different tests independent of each other using two completely different data sets, and we seem to be getting a fairly consistent signal. But whereas marine forms never seem to recover from this crash, terrestrial taxa do rapidly recover and radiate into lots of new groups, including crown group crocodilians seem to originate around this time, as well as things like Notosuchia, an important terrestrial group, kind of like little crocodile tanks which used to walk around on land and even were herbivorous sometimes. They're very cool. The environmental candidates which we've selected for these patterns seem to be sea level changes, sulfur toxicity, and large-scale kind of tectonics, as we might expect. But we can't rule out that things like bolide impacts were important as well. So everybody knows about the kind of Chicxulub crater wiping out the dinosaurs, yada yada. It's a really boring story. At the Jurassic-Cretaceous boundary, we actually have 3 bolide impacts, which for some reason no one's ever really decided to look at. And preliminary data actually suggests that one of these bolide impacts was actually about 50 kilometers wider than the Chicxulub impact. So these guys could have played a role, but it's very difficult to associate a singular event with a kind of long-term trend because we can't support it statistically. So we can kind of speculate that maybe at some point these extraterrestrial impacts are having an impact. So the next role, we've already heard about the Red Queen Court Jester issue. Is it one or the other? I don't like to think about it like that. I like to think that they both work together through time to drive macroevolution patterns. So my next thing is using this big new super tree to look at phenotypic evolution through time and seeing if we can find kind of functional changes through time which might correspond with environmental changes too, and drove these cool patterns we're seeing in crocs. And I'll happily take any questions about that.